Right everybody, I'm going to take you through the sort of short and simple version of making a smear, which is the first stage in staining bacteria. So here we've got our bacterial culture, in this case it is Bacillus subtilis. We've got our cleaned, so I've cleaned this microscope slide with a lens tissue just to make sure it's nice and clean for our use, not to get any weird artifacting on the lens or in your vision when you look through a microscope at it. It's labelled with the species, Bacterial or Bacillus subtilis, the date of which you've performed the experiment, and my initials, in this case CD. We've got our metal wire loop, which I'm going to be flaming a few times, as you'll see. That's for transferring bacteria across and spreading stuff along the slide. And we've got just a beaker of tap water, just so we can make our smear. So, first thing we're going to do is we're going to flame, so just hold the metal loop in the flame, and because it's got water on it, go back to the water to evaporate first. So you need to get it nice and glowing. Nice and glowing. And let it cool for a bit. And then you're going to transfer 2 loopfuls, so just sort of loopfuls of water onto the centre of the slide. So just leave it there, a little bit of water, and you can spread it around. So it's a nice sort of maybe 1 centimetre, 15mm diameter, sort of a blob if you will. I'm going to flame the loop again, just do that at every opportunity. Nice aseptic technique. Right, so that's that. And now what we're going to do, we're going to open our bacterial culture. I'm going to try our best to do aseptic technique here, which in this case is retaining the lid on your hand. I'm going to flame the neck of the bottle just to make sure any sort of falling debris in terms of cellular stuff will be killed. And we're just going to gently, I'm not going to be able to see it, the focus is a bit dodgy, just touch a colony. I'm not going to take too much, just make sure it touches. So I'm going to do that one. Alright, so that's touched it. So now, before we do anything, I'm just going to return the lid to that culture bottle. It's going to stay there. And now what we're going to do is we're going to take what little bacteria there are on this loop. I'm not sure you'll be able to see it. If you can see too much of it, it's too much. You only need a small amount. We're trying to dilute that cell concentration to something visible but not sort of overlapping. So we're just going to find that, you might be able to see the little sort of blob of water, and we're just going to move it around, swell it up a bit, spread it out a little bit wider to about 2 centimetre diameter, so it might be easier to see. It's kind of hard to see the liquid present, but it is here. Might be, if I tilt it in the light, you might be able to see it a bit more. So it's just spreading it around, mixing it in about a 2 centimetre area ish. And once you've done that, you then again sterilise your loop, burning it in the flame, nice and red hot. I'm just going to cool it down in this tap water again. Right. Now what we're going to do is, just to make sure this is nice and dry, we're just going to gently hold it in and out of the sort of funnel of heat, just so it dries off a bit. In and out. So it might be, I might have to lift this out of view, but I'm just going to dry it up so that that little spot of water is gradually receding. It's gradually receding. Obviously be careful you don't burn yourself, and you can see, well, you might not be able to see actually, but there's no visible droplets of water left, so that's now dry, but we need to properly heat it. So we're going to dry the suspension by fixing it, which means just getting it to the sort of apex of the flame, sort of the hottest part in the sort of centre of the Bunsen burner flame, and just wafting it through a couple of times just to really fix the suspension to the surface of the slide. And that's that. So I'm not sure you'll be able to see it, but you might be able to see that there's a sort of milky white layer on top of our slide, which is our suspension. Anyway, I'm not quite sure if it'll pick up very well on camera, but you might see it just about there. There's a milky white suspension, and that's our heat fixed smear, all ready for staining. Right, on to the next one.